Advances in assisted reproduction. What can we expect? This discussion event has been made possible thanks to the support of the Scottish Government, and we at PET are extremely grateful to them. So we have 4 professors speaking this evening. That's Rod Mitchell, Evelyn Telfer, Adèle Marston, and David Albertini. And Rod is Professor of Developmental Endocrinology at the University of Edinburgh and at the Medical Research Council's Centre for Reproductive Health.
So thank you very much to the PET team and to the sponsors for allowing me to present to you today on preserving fertility in young males. And I'm going to talk to you a little bit about what we think the future might hold. So I'm sure many of you will be aware that fertility in males requires sperm production, but that sperm production only starts from mid to late puberty and continues then on into adulthood. So whilst obtaining sperm from men who are able to make sperm might be an option for fertility preservation, for those who don't make sperm, and particularly for children, this is not an option. So what do we do in the situation where we have individuals that can't make sperm? And when we think about the children, then really the largest population that are affected in this way are those who aren't going to be able to be fertile as a result of treatment that they've received during childhood. And this is primarily those that are receiving chemotherapy or radiotherapy, which we know in some situations can affect future fertility. So children with cancer or children with other diagnoses that require these types of treatments could be at risk from future infertility. Now, I've mentioned that these individuals will not be making sperm yet, but there is something that might be able to be preserved to protect and preserve their fertility potential. And this is spermatogonial stem cells, or SSCs. And these spermatogonial stem cells are cells that are present in the testis at all stages of life, so they're present in children as well. And these cells we know have the capability to make sperm, but don't yet do this until adulthood. So is there a way that we can preserve and save these spermatogonial stem cells from individuals that we know are at risk of future infertility? And the way in which we've started to do this is to obtain small pieces of testicular tissue, so to do a surgical biopsy, obtain a small piece of tissue, and then cut that piece of tissue into fragments, place it into freezing straws, and put them in the freezer. And this essentially means that we can preserve these stem cells for as long as these individuals might potentially want them for use in the future. So we can preserve the spermatogonial stem cells, but how are we actually able to achieve this in terms of offering this to patients? So this is what we are currently able to do in the UK. We have established our service back in 2015 in Edinburgh. There is a second centre in Oxford that are coordinating these testicular biopsies to preserve the spermatogonial stem cells. And the key thing is that this is an expanding service, which is now offering this to patients across the UK. But we are still looking to make sure that we are providing good access for patients to receive this procedure. We've now done this for more than 350 patients over the last 5 or 6 years who are in childhood, and these tissues are sitting in the freezer ready for them to use in the future. But what can we actually do with the tissue at the other end? So here you can see an example of some of the things that we might be able to do with this tissue once these individuals come back to us asking for their fertility to be restored. So the tissue can be taken from the patient, can be frozen. When they come back, we might have the option of taking the cells of the testis, spermatogonial stem cells, and injecting them directly into the tubes of the testis, such that that repopulates the testis with these stem cells, and that could then generate natural fertility again. Alternatively, instead of injecting cells back into the testis, we might be able to take tissue pieces, graft them into the testis, and then generate sperm in those transplants, extract the sperm, and use those for artificial reproductive technologies such as IVF or ICSI. If it's not possible to transplant the cells or tissue back to the patient, The third option that we might have is in vitro maturation. So essentially try to grow these sperm in a dish. So take the tissue, allow it to mature, and then obtain those sperm. And again, if we can do this, this could be used for IVF or for ICSI and artificial reproductive technologies. So these are the potential options of what we might be able to do with these stored tissues and stem cells. But how close are we to being able to do it? And I would say we're really now getting quite close to being able to offer a genuine clinical option to patients. And that comes from some of the preliminary data and the provisional studies that we have in the primate model. So the primate or monkeys are obviously animals that are very close to humans. And we're now able to show a number of studies have done transplant approaches where they've taken tissue from a prepubertal monkey, frozen that tissue, taken the tissue out and thawed it after the individuals had treatment, and then to put the tissue back into the testis to generate sperm. And these sperm are then potentially available to be able to create offspring. Now, this is obviously a primate model, and this is not proof of principle for being able to actually generate offspring. And the real breakthrough in this area came relatively recently, where instead of just simply being able to produce sperm by transplanting tissue in the primate model, what our friends and colleagues in the United States were able to do was take this one step further. So they took the sperm that were generated from the monkey testis and have been frozen. They then injected those sperm into an egg. They generated embryos, viable pregnancy, and then we saw relatively recently the first live offspring from a transplanted testicular tissue from which we'd obtained sperm. And this is a proof of principle in the primate that the whole process from obtaining tissue, storing stem cells, generating sperm, and being able to generate offspring with those sperm was feasible. So I think we're very close now to a situation where we're actually going to be doing transplantation of testicular tissue back to patients after they've had their treatment. And there are a number of centres who are on the cusp of offering this, obviously as part of a clinical trial, very well controlled clinical trial. But I just want to really highlight one other aspect of fertility preservation, particularly in the young patients, and that is in these patients who are in puberty. And I often would refer to these as the in-betweeners, not necessarily because we would all aspire to be like the characters in the in-betweeners, but the term the in-betweeners is absolutely perfect to describe the situation we have in terms of fertility preservation in adolescents. And that is because these adolescents are somewhere in between a situation where we might want to obtain somatogonial stem cells, i.e. there are no sperm there to collect, or these individuals may be in a situation where there could be sperm present. And in this case, we would be wanting to store semen or produce or undertake a surgical option to extract sperm from the testis. And because these individuals are somewhere in between the two processes, these individuals are often sometimes overlooked when it comes to offering them fertility preservation. It's not appropriate to offer a testicular biopsy to store spermatogonial stem cells in a patient where we know they might have sperm. But equally, in many individuals, particularly those who are under the age of 18 and are in the middle of puberty, the provision for semen cryopreservation is variable, and the option of testicular sperm extraction is rarely offered in the under 18 age group. So whilst I think we are offering a good service to the adults in terms of semen cryopreservation, we're developing these options for the prepubertal patients, I think the adolescent boys are still a group that we need to ensure that we can provide them with a better service. So in terms of summarising what I've told you, testicular tissue cryopreservation is certainly an option for young males at risk of infertility, e.g. those who are going to have cancer treatment. We know that retransplantation of testicular tissue is very close now to being tested in clinical trials, and we hope that in the not too distant future, we will see some successes from this. that improving provision for the adolescent males should really be a priority for our fertility preservation services going forward, and that the future options for fertility preservation may not in fact require germ cells at all. And whilst I haven't talked about that in this talk, this might be something to discuss in the questions, in that there are now research studies in animals which are looking at ways in which we might be able to generate sperm without having to have a germ cell as a starting point.
Evelyn is a professor of reproductive biology at the University of Edinburgh and leader of the first research into the full development in human eggs, which was achieved in the laboratory.
Thank you very much, Sarah. So Rod has spoken about the male system, and we have some differences, of course, in the female system. And I'm going to talk about the fertility preservation options in females and some of the new developments that we've been involved with. So females are born with all the eggs that they will have. Under normal circumstances, that would be the case. And the eggs that are formed before birth, so during fetal life, have to last the woman or the female throughout her reproductive lifespan. So there's the eggs are being formed, and then by the time of birth, they're actually starting to be lost, so they're degenerating. So with time, there's a gradual loss of eggs with age. That occurs quite sharply after 30. And the decrease in the number of eggs, which determines the fertile lifespan of the woman, can be accelerated under certain conditions. So with some chemotherapeutic regimes and radiotherapy, there can be a more dramatic loss of eggs. So what can happen then to preserve fertility? So you have the resting eggs, what we call the primordial follicles, the immature eggs. Actually, 99.1% of those eggs will end up degenerating. They die. Only 0.1% will ever be ovulated. So the question is, can we save these eggs and can we preserve fertility in the female? So fertility preservation is a field that has expanded because of the work that's been carried out in Edinburgh by Roger Gosden and David Baird, who developed the freezing technique for ovarian tissue, firstly in sheep, and then it was rapidly translated to humans. So in the 1990s, Edinburgh started a clinical storage program for ovarian tissue. So ovarian biopsies can be taken, and that tissue will contain the immature eggs. And this is the only option that prepubertal girls have to preserve their fertility. Clearly, adults have other options because they can freeze eggs or perhaps embryos, but for young girls, it's really only the storage of tissue. And the tissue then that's been frozen can then be thawed when the woman would like to have it used or utilized. And those strips, so this is showing you some of the strips, the ovarian strips, they contain hundreds or thousands of immature eggs. And the options available at the moment are for those strips to be transplanted back to the site where the ovary is. And worldwide, there's been over 130 babies born using ovarian transplantation. So that's a good ovarian fertility preservation option, but it's not suitable for everyone, because many of the women who are going to fertility preservation have blood-borne cancers, and these cancers may result in malignant cells being present within the ovarian tissue. And of course, you would never transplant it back because you could then have a recurrence of the cancer. So what would the options be for those patients? So this is the area that we've been working on, where we take this ovarian tissue and we then grow it entirely outside the body. So we make lab-grown eggs from the ovarian tissue, and this would avoid the transmission of malignant cells, but could also be used in a whole range of patients. I'll come to that later. So this ovarian tissue then that's been cryopreserved and stored contains predominantly immature eggs. And the challenge that we have is growing them from this very immature stage to the point where they can be matured and fertilized and hopefully form embryos. Now, we can do this in mouse, and this was carried out in the early 1990s in mouse in the US at the Jackson lab. And this is the first mouse that was born after its egg was grown entirely outside the body from the most immature stage. This is egg birth. Now, many embryos can be formed and offspring produced. So, but what can we do in the human? Because translating from mouse to human is incredibly difficult. The organs are really completely different. Although we're dealing with a similar process, it's a very, very different structure, and it requires a different environment. So where are we then with growing human eggs outside the body? So we've developed a multi-step culture system to grow these immature eggs. And we start with this tissue piece, human ovarian tissue. And we obtain the tissue through informed consent, and women donate this tissue to us. And then we can activate the resting follicles, these immature eggs. We can activate them to grow, and you can see them starting to grow in the lab. And then with the growing ones, we can isolate those growing ones, take them to the next step, get them to develop further. So we isolate them with their surrounding cells, and the egg is in there. And then we remove the egg. This structure is the egg with its surrounding cells. We can remove that structure, and then we can place it on membranes to push them down to the process of resuming meiosis, which Adèle will speak about in more detail. And what we can achieve then is oocytes that have been grown all the way from the most immature stage, we can get them to reach this point of maturity. And this is quite similar to how they might be at the time of ovulation. So they've resumed meiosis. And anyone who knows about eggs would know that these look rather strange, but we've been modifying our techniques, and we get more morphologically normal eggs now. So we've changed some of the physical conditions that we grow them in, and we can get these human eggs grow to maturity entirely outside the body. But the next step, of course, is to test the fertilization potential and embryo development, and to determine whether that indeed is normal. So we have ongoing experiments to test the safety of these lab-grown eggs, and we're currently using a sheep model in collaboration with people, Helen Picton at the University of Leeds and other groups within Edinburgh. So hopefully next year we'll have more information about the fertilization potential of the sheep lab-grown eggs, and obviously we hope that we'll be able to obtain lambs from those. And of course, at the same time, we're optimizing our human system, but we're also adapting our human system for different patient groups. So we've mainly been working on healthy women, tissue from healthy women, but we're now looking at how we can improve the system for pediatric tissue, for tissue that comes from transgender population, and Turner syndrome patients. So all of this tissue is different and will require adaptations to the culture system. So in the future then, this process of lab-grown eggs, or as we refer to as in vitro growth, could replace conventional in vitro fertilization. And there's advantages to that because it bypasses the hormonal stimulation that is required for the cycles of IVF to retrieve eggs. So that could be a future application as well as for fertility preservation. But another part of the future, which I think you'll hear some more of from David Albertini, is that we could use this system when we're making new eggs from stem cells and then growing them to maturity in the lab. I'll just finish very briefly by saying some of the work that we have going on in the lab is that we're taking human ovarian stem cells, we're mixing those with support cells, we're forming these mini ovary structures. And within these mini ovary structures, we have what appear to be structures like eggs being formed within them. So it looks like new eggs are appearing within these aggregates. But of course, a lot of work has to be done to show that these are in fact eggs and that they're normal eggs. So stem cells and lab-grown eggs open up new possibilities for fertility preservation and the treatment of infertility, and they would provide options for a wider group of patients. And remember that these may be applicable to the young girls who are storing tissue now, because we have patients as young as 18 months old that are storing tissue. So as Sarah said, when we looked at the coloured television, this could be something way into the future that will be clinically applied. So thank you very much.
So I'm going to turn now to our next speaker this evening. And next up, we have Adèle Marston. And Adèle is Professor of Cell Biology at the University of Edinburgh and a Senior Research Fellow at Wellcome Centre for Cell Biology.
Thank you to Sarah for the introduction, and thank you very much to PET in general for inviting me to give this talk today, and also thanks to Rod and Evelyn for some very enjoyable talks. So as we know, assisted reproduction gives many people the opportunity to be parents, but it does not have a perfect success rate. And the reasons for this are complicated and unclear, but a major cause of infertility is eggs, so maternal reproduction with the wrong number of chromosomes. And this is a condition known as aneuploidy. And it's thought that up to about 30 or 40% of all human eggs are aneuploid and have the wrong number of chromosomes, while about 1 to 2% of sperm have the wrong number of chromosomes. And of course, if you have an aneuploid egg or an aneuploid sperm, you will generate an aneuploid embryo. And most aneuploid embryos cannot develop, and this results in miscarriages. But there are a few exceptions, and the most famous of these is Down syndrome, where individuals have an extra copy of chromosome 21, so 3 copies rather than the normal 2 copies. And as I've said, we know that the major cause of aneuploidy in embryos are errors in the egg. And that is during the process of meiosis, which is the cell division that sorts the chromosomes into the mature egg. And we also know that this aneuploidy increases as women age, and this increase in aneuploidy as women age is recapitulated as an increase in miscarriages as women age. So by the time a woman is in her 40s, she has about a 40% chance of a miscarriage, and this increases exponentially thereafter. As I said, this aneuploidy arises due to errors in meiosis, and during meiosis, the chromosomes need to be sorted into the mature egg. And as Evelyn explained to you, when a girl is born, she is born with all of her immature oocytes or eggs, and these need to develop at puberty. And in the immature eggs, the chromosomes are arrested in this prophase 1 state. And what this means is the chromosomes are already assembled in a configuration that makes them ready to undergo this sorting process. And indeed, they also are thought to have a lot of the key proteins and regulators already assembled on them, which will then need to be maintained throughout the life of the woman and the reproductive life of a woman. So you can imagine that some of these proteins might need to exist and be stable for 40 years. And it's thought that deterioration of these proteins is actually one of the causes of infertility. Because if you lose these proteins, then the chromosomes will not be held properly in this configuration, and this sorting doesn't happen properly. So during puberty, the oocyte will grow and then resume meiosis and go into the meiotic divisions. And the first meiotic division will result in the extrusion of this polar body, which essentially throws away half of the chromosomes, but a very specific half of the chromosomes. And this is very important, and this needs to be controlled very carefully. And this egg will now wait for fertilization by a sperm, which will trigger the extrusion of a second polar body, again, in a very specific and a defined way. And so that you end up with the embryo with these two pronuclei, one from the mother and one from the father. And so all of this requires some really quite complicated acrobatics on the part of the chromosomes. So this is illustrated here for one chromosome. You can see that you have various crossing over events, segregation, and 2 consecutive segregation events, so that we end up with exactly the right number of chromosomes in the mature egg. And so our interest in the lab is to try and understand what the molecules are that direct these specialized acrobatics to ensure that we have the right number of chromosomes in the eggs, and therefore not no aneuploidy in the embryo. Over the past few years, we have work in our lab and work from many other labs have provided a working model for how this happens. And this is mainly coming from studies in model organisms such as yeast and frogs and mouse. And more recently, we've been trying to ask the questions of whether what we've discovered in model organisms is actually true for humans. And if that is the case, what is it that's actually going wrong with these pathways? And this is a project that we've initiated in the last about 3 years. And what we're doing is we are gratefully receiving donations of eggs and embryos that cannot be used for treatment that have been donated by patients. And we receive material both from IVF and ICSI regimes. And in IVF, we get eggs which fail to perform pronuclei or have abnormal pronuclei and therefore are not suitable for treatment. From ICSI, we generally get eggs that have not been injected, usually because they are not mature enough. And so what we end up with is a whole range of eggs actually at very different stages, and we never quite know what we're going to get. And so we have to think of different stages in the process to analyze these eggs. But one of the key questions that we really want to understand, it has been developed from our understanding of model organisms. So as I explained to you, early in prophase 1, the chromosomes have already adopted this configuration in the immature oocyte, which then will need to be maintained throughout life. And that looks schematically something like this, at least from what we understand from model organisms. And this whole structure needs to be properly held together by various different molecules or proteins. We need the kinetical, so in model organisms, we know that kineticals, which are these structures that assemble once on each chromosome, are actually fused together to hold these 2 chromosomes together. We know that this is not the case in humans, and we want to understand molecularly why that is the case. We know that cohesion is present and holds the chromosomes together. And so we're also asking whether deterioration of cohesin and another protein that regulates it called sugoshin could also be a factor in human fertility. So it's early days. We're still asking whether these factors are actually even existing in humans and whether they're functioning properly. And I'm just going to show you one example. So here's a human egg in meiosis 1. And you can see here that the chromosomes take up a very small part of this egg. But if we are to zoom in on them, we can see all the chromosomes here clustered together. And in our lab, we're mostly using fluorescence microscopy techniques to light up particular structures. And if we just zoom in here on one of these single chromosomes, you can see that here we have the kinetochores labeled in red and this cohesin regulator in green. And as has been shown by others previously, these kinetochores are visible as two separate foci, suggesting that they're not fused. But interestingly, this Shigoshin cohesin regulator can be seen sort of around these kinetochores, but also between them, which would put it in exactly the right place to actually be holding these chromosomes together. So this is just one example. Obviously, we need to look at a lot more examples and before we can really generate any conclusions. But I hope that this will be promising to try and understand the underlying molecular causes of aneuploidy. And so to go back to the question at the beginning, so what can we expect for advances in assisted reproduction from our work? Well, what we hope is that we can understand more about the origin of human aneuploidy and therefore hopefully eventually understand what goes wrong, and then use this as a tool in diagnostics to provide much better choices for patients in their treatments. And just to thank the people involved, so this is part of a very large collaboration between the University of Edinburgh and the University of Warwick, with my colleagues Evelyn Telfer, Richard Anderson in Edinburgh, Andrew McKinch, Geraldine Hartshorn, and Nigel Burrows in Warwick. I won't read out everybody's name, but I particularly want to thank Bettina Mahalas, who has been doing this work in my lab, Norma Forson for the research nurse who has been helping with the patient recruitment, especially the patients that have generously donated all their samples, and the embryologists and clinicians at EFREC that have been really helpful in letting us come into the clinic and get the donations. And thank you for your attention.
And finally this evening, we have David Albertini. And David is Professor of Developmental Cell Biology at the Bedford Research Foundation and visiting senior scientist at the Rockefeller University and at the Centre for Human Reproduction. And David is also Editor-in-Chief of the Journal of Assisted Reproduction and Genetics.
I'm going to go over really some big picture material. I think what you've heard from our speakers so far, both Rod and Evelyn, have given a very nice up-to-date coverage of how this field of fertility preservation has evolved over the years. And in fact, I think it would be fair to say that, that assisted reproductive technology has really enabled the field of fertility preservation to take the form that it has now, where we're really offering to patients, specifically cancer patients, an opportunity to reproduce, whereas in the past, this was simply not possible. So what I'd like to do is sort of view the field of assisted reproduction from a bit of a historical perspective first, and suggest that before 2010, we saw many of the fundamental discoveries that came from the use of model organisms, as Adèle has just referred to. Before that time, we built on that foundational knowledge in basic reproductive biology. And took that knowledge. This is the bench to bedside transition, and we moved it to patients. And this was really the foundational work of Bob Edwards when he started this field. And it took over for the next 20 years, or actually almost 30 years. We saw, as this graph suggests to you, at least up to 2011, we saw an enormous increase in the adoption of assisted reproductive technologies to aid primarily patients who were presenting with various kinds of infertility. Of course, it was back in the 1990s that the technique of intracytoplasmic sperm injection, or ICSI, was introduced, and this itself was a huge boon to the process of baby making by ARTs, because so many of the eggs that we attempted to do, let's call normal fertilization before that, were not fertilized. And so once ICSI came on board, there was a huge explosion in the availability of this technology for patients who needed treatment for infertility. And, really pretty much up to 2011, in fact, when we look globally now at the number of, in this particular graph, the number of cycles that people were undergoing for treatment of their infertility, there was this steady rise in the technology. And since 2010, we've seen a number of new what the HFEA calls add-ons enter into the practice of assisted reproductive technologies. And in alignment with the expansion, if you will, of the technical options for patients to choose, came a change in this bench to bedside movement, because the science of basic human reproduction has now very much moved into the realm of big business. Okay. And this, as you might expect, has been accompanied by a huge increase, not only in the numbers of patients seeking traditional ART treatments, but, for example, and fertility preservation, as we've just heard. But, for example, as this graphic shows you below, and as apparently your last session was covering, the whole topic of egg freezing has made it possible for women to store their eggs, again, at a younger age, relevant to the point that Adèle made about the impact of age. And this business has essentially expanded well beyond its original intentions, which were to treat fertility. And, this shouldn't be too much of a surprise. We have a history in the field of reproductive biology of breaking new technologies very rapidly into a clinical application before, in some cases, really thoroughly evaluating not only the efficiency, which I'll come back to, but the safety of these procedures. And so we've always understood that in traditional ART, women are subjected to injections of powerful hormones that will stimulate the development of follicles. And there's always been concern about the long-term consequences of this kind of treatment. It's known as controlled ovarian stimulation on the health of women down the road, at advanced stages. But of course, what has come along with the popularity of this technology is the availability to, first of all, make stem cells. This we realized in the early 2000s. And not only the ability to make stem cells out of embryos that were donated for this, that, or the other reason, but also now we're in the midst of a discussion of other things that have been linked to the practice of advanced reproductive technologies. I'll just mention a couple here. That this past May, the International Society for Stem Cell Research released their latest guidelines over gene editing. Okay, And to many, especially those in the ethical community, this in fact addressed the whole issue of how long should we be culturing human embryos in vitro. And you know, if I can, I'll just mention that this image here came from a 13-day human embryo. It came from the Brunello laboratory at Rockefeller University. And this and work of others showed us that, in fact, the human embryo could develop on its own. It had a certain amount of autonomy, okay? I think the conversation that we're having today about where this technology would go is a timely one and long overdue. And if you follow kind of news coverage of some of the advances we're now going to talk about, you know, flashing back to the time that Dolly appeared on the map, we also see a growing sense from the public of mistrust in the scientific community, which I think invariably we can associate with the commercialization of this process. And some would go on to say, as I'll come back to, as this ethicist has noted, the selective breeding in the era of reproductive technologies is with us. So the fertility market is booming, and along with it has come a parallel expansion in the growth of new genetic technologies, okay? Technologies that are suggesting, if you want to take the gene editing concept to heart, that someday we may be able to extend human lifespan, okay? That we may be able to manipulate not only embryos so that disease risks are minimized, okay? But that there could be enhancement of various properties, okay? And here we have, you know, a recent company offering to select the genetically perfect children from a cohort of embryos that is now well within the realm of possibility with some of these new genetic testing technologies. So where are we going with the field of assisted reproduction? We've already heard from Evelyn that there is much discussion now based on the possibility that stem cells, specifically iPS cells or embryonic stem cells, could be used as starting material to generate human sperm or human eggs. And as already alluded to, the work of Hayashi in Japan using mice has shown that this is a very real possibility, that sperm or eggs can be made. They can be used to make embryos, again, mouse embryos, and that these mouse embryos give rise to live young. We're also poised to enter into the gene editing era with not only the advances that resulted in the Nobel Prize a couple of years ago in the CRISPR-Cas9, the power of the CRISPR-Cas9 gene editing, but now we have base editing, we have a number of new technologies and new variants of the CRISPR technology that have added precision to this genetic splicing that has been shown to work in various kinds of cells and will continue to be explored as a very powerful technology. Just briefly coming back to the matter of egg aging. Again, as Adèle pointed out, the incidence of aneuploidies and miscarriages increases with progressive maternal age. And one of the reasons for the loss of egg quality and the errors in chromosome segregation has been the thought that mitochondria in the egg can no longer perform the energy demanding role that they play. And so now we've heard of three-parent embryos, embryos where the eggs the eggs carrying the presumed deficient mitochondria might be replaced by healthy mitochondria. And then with the union of the sperm, we would have individuals who would be comprised of genetic material from 3 individuals. And of course, now we're in the midst of trying to sort out where lies the hype. The hopes are certain for people in different categories, and we're certainly moving on a very slippery slope into the future. So, in summary, I think we need to look very seriously at the positive side, of course, is that there are already over 8 million babies. We're marching towards 10 million babies that ARTs have much to be proud of contributing to, and those families that have been made possible. Along with this growth, especially as we see it in the United States, has come a free market environment that lacks any and all regulation. Okay, and so that opens the door for misuse and abuse with these technologies. The constraints of commercialization have also been shown, again, especially in the United States, to contribute to restricted access of this technology. Low and middle class members of society do not have access to these technologies in countries like the United States, where there is not mandated coverage for that treatment. So indeed, it's time to think anew about the future of this technology that's helped so many, but has the potential to harm many as well.
Presumably, if testis, ovary tissue can be preserved, stored or used from prepubescent children, it will be able to be stored for 55 years because it's for medical reasons, so it's covered by the current law. Does that mean that the child, male or female, can use the eggs or sperm to create a baby up to age 55 or even 67, say, with a 12-year-old boy? So that's the first bit of the question. Then secondly, do the panel think that such an advanced age to have a child is ethically, morally, medically justifiable? And how does this accord with Section 13(5) of the Human Fertilisation and Embryology Act? So, Evelyn, do you want to talk about that first, and then perhaps Rod?
Yeah, sure. Yeah, well, in theory, yes. If the tissue was thawed, 50 years ago, I mean, of course, there's no precedent. We really don't know whether the tissue would survive that length of time in freezing. We do know that tissue has been, frozen for a substantial period of time, almost 20 years. I don't know offhand what the longest one is that's been used, but I think the pregnancy that was obtained from prepubertal tissue was around 15 years that the tissue had been frozen. So 55 years, yeah, is a long time. But I think realistically that would not happen because it would be dependent on the desires of the person who the tissue belonged to. Now, do I think it's ethical to use the tissue after that length of time? I personally do not think that someone who's 50, well over 55, I think most people don't really want to be starting to have children at that age. Of course, there's a debate to be had about storage, but I think storage will or should always be longer than we would expect to use it. But yeah, I mean, it clearly opens up a lot of ethical issues that have to be discussed and have to be placed in the context of how we regulate things within the delivery of assisted reproductive technologies. So within the context of the HFE, and other countries have to deal with their regulatory bodies as well.
Rod, do you want to come in on this in relation to male factor?
Yeah, yeah. I think the first thing to say is that the amount of time that the tissue can be stored for is based more on making sure that that tissue is available at the time at which the patient would need it. So, for example, a 1-year-old, a 2-year-old, and we do this in patients as young as that, is unlikely to be coming back for at least 20 years. So we have to have a relatively long period of time over which this tissue is available for future use. And 55 years is what the tissue that we store is currently able to be used for. I think the debate in terms of the ethics of the age of parents and things like that, we maybe need to think about the males and females slightly separately, because certainly we hear enough about very elderly fathers having children naturally. So for males, this situation might be slightly different in terms of the fact that males can reproduce naturally into a relatively old age. So maybe that's something to also think about.
And David, you very much talked about the ethics in your presentation. Do you want to come in on this about, the advanced age to have a child and the ethics of that?
Sure. I agree with Rod that, there are two very different sides to the coin here when it comes to gender. The clinic that I work in New York City specifically deals with older women. And there is no question that there are health risks that a woman takes on with advanced maternal age. We have many women who are in their 40s who come to the clinic. Unfortunately, their own ovaries are not able to produce many very good eggs, so we don't get good embryos to transfer for those women. So they generally go to an egg donor cycle. So they'll receive eggs from a younger patient, and up to the age of, I think the oldest patient, and we're only one of 2 clinics in the United States that specifically deal with the older female patient. The older patients that I've seen who have carried babies to term, 46, 48 years old, I think is the time range, are generally healthy, but there are many more pregnancy complications that come along with carrying a child at that age. So that's I think that's a very, very significant issue that plays into ethical choices. I don't know, does the HFEA have, what is the age guideline for the UK?
It's that there isn't one.
Okay.
The clinics self-regulate on that, and
right.
we don't hear about women. Well, very rarely we hear about women over 50 being treated, and over 50, I think there's perhaps been one in all the time. And I think it's always important to remember that the doctor treating the patient has a duty to that woman if he's going to be transferring an embryo into her for her health. And that is obviously, it's very, very important. But there's a follow-up question really from that. From somebody who did her research at Edinburgh, and she says that she's 48 years old and still having periods, but her ovarian reserve is very low. She wants to do ideally IVF with her own eggs, and is it possible to imagine to regenerate and even increase the number of initial eggs? I think she's asking about her own there. I don't know who wants to go first on that. Perhaps David and then Evelyn.
So there is a technology being studied right now, which involves the injection of the human embryo with, it's essentially an extract of growth factors from platelets. This PRP technology has been used to revitalize various organs over the years. And so, for example, for joint health, it seems to actually work. But I can tell you, because through the journal, we've seen more studies of this nature coming through in the last 4 to 5 years, that at this point, it doesn't look very promising. Okay, and the idea is not so much that it would stimulate the formation of new eggs. Okay, this woman knows that her ovarian reserve is low already, but the idea is that it would create a more favorable environment in the ovary for the follicles to develop, after she receives stimulation. So other than that approach, which is not looking promising at this point, I don't really know of any other approaches to the problem.
Okay. Evelyn, is there anything you want to add there?
Yeah, I mean, I would agree with David. I mean, there's a lot of preliminary studies that are looking at ways of boosting primordial follicles. I think the questioner is perhaps alluding to the work that's been done on oogonial stem cells, so trying to induce stem cells that are within the adult ovary to make new eggs. And that, of course, is work that's ongoing in a range of species. But what we don't understand is how you might do this within an ovary that's close to, depletion, that's menopausal, because the premenopausal ovary is quite a different landscape from, the sort of, younger stage of ovary, and it changes. So I think we've got a lot to learn about the different cell types that are there, how the ovary will respond to, other cell types. And so I think realistically, there's nothing at the moment that would regenerate eggs in an older woman.
Thank you. Our next question is for Adèle. And so Adèle, the cryopreservation of sperm and ovarian tissue is now largely used for fertility preservation purposes. Does the cryopreservation process have an impact on chromosome stability and meiosis in eggs frozen with ovarian tissue or in frozen sperm? I don't know whether you've come across this in your research.
But this is a great question. Thanks for this. So this is actually something that Evelyn and I together are very interested in looking at. So in Evelyn's system, where they are growing eggs in the lab, we don't actually know what state the chromosomes are in at the end of that, and that's something we plan to look at. But I think the problem that we have in doing that is actually we don't really know what normal is, because we still have a lot that we just don't know. So as I said, we're looking at this in the eggs from the clinic, and we're also looking at this with people in Warwick with some sort of more healthy donated eggs. And then we're also looking at this with the cryosections from Evelyn. So I think when we have all this information together, then we can sort of make those conclusions. But it's a great question, I think. But yeah, I'm afraid I don't have the answer. I don't know if Evelyn wants to add anything, but
no, I think the cryopreservation question, I mean, it is a great question. And what we have to remember is that the way tissue is frozen, there's not really been many advances from the work that was carried out by Roger Gosden in the early 1990s. So I think there's a lot of work that can be done or should be done with, the freezing techniques, and also to determine whether it does have any impact on the developmental potential of the tissue.
And perhaps people who have their eggs and sperm in storage but know they're not going to use them, perhaps that would be a useful avenue of research that they could donate those to. So I'm going to move on to the next question now, which is from Natalie Sutherland, and I think Rod, if you could answer this first, please, because we sort of saw some images on your slides. How invasive is the procedure for extracting tissue from young boys and girls? So
for the boys, it's relatively minimally invasive. So because the testicle is just under the skin of the scrotum, it's quite easy to make a small incision pull the testis out through that hole and make a biopsy of the testis. So it's relatively minimally invasive. For the females, it's a little bit more invasive because it requires actually going in into the more into the body than perhaps compared to the testicle. So it is more invasive in females, and I don't know if anyone else wants to comment on the female side for the biopsies. It tends to be done, it can be done laparoscopically, so keyhole surgery, which is obviously less invasive than an open approach, but it is more invasive than the male. I think it's probably the easiest way for me to say it anyway.
Evelyn or David, did you want to come in on that at all? I think.
Well, it still requires laparoscopy. I mean, to my knowledge, I mean, you could do, as you know, you do transvaginal aspiration for normal egg retrieval, and it is possible to get small biopsies that route. But in general, correct me if I'm wrong, Evelyn, but in general, or Rod, you know, especially these young patients, the strategy, as I understood it in the early days of fertility preservation, was for the women, for the girls, to remove one ovary in its entirety and leave the other one in, because it's always difficult to anticipate whatever consequences of radiotherapy or chemotherapy are going to be. Because in fact, some patients recover just fine and don't experience gonadal toxicity. But once you have a whole ovary out, then you can process it into all of the strips that Evelyn mentioned.
Yeah, I mean, that's a method. Edinburgh does not do that and has not done that. Edinburgh takes pieces of tissue and stores, but in Denmark, so the work Klaus Andersen, they always remove one ovary, so they have the whole ovary then to store. But yeah, all the patients that I'm aware of have had, apart from removing one ovary, it's been ovarian biopsies via a laparoscopic surgery.
Okay. Yeah, it's also worth probably just adding to the end of that, Sarah, that the complication rate for both testicular and ovarian cryopreservation is very low. So, if there's a question about which is more invasive than the other, perhaps we should be just thinking about it from the point of view of the possibility of complications. And for both, it is low. So,
yeah, obviously, particularly in this patient cohort, probably more than any other, that safety element is so important. So I'm going to move on to the next question, which is from Andrew Lockley. Can sperm creation cells be obtained from women or girls? So I don't know who wants to go answer that one.
I think the short answer is no. but Rod.
I think probably, it's probably easier to go with the short answer of no. I think if we're saying here, can sperm be created from an ovary, and or vice versa, can eggs be created from a testis, then the answer is really no to that. David did allude to the idea that you might be able to generate sperm or eggs from cells that aren't germ cells, i.e. they're not oocytes or spermatogonial stem cells or any of the germ cell kind of lineages. So in future, it might be the case that you can generate germ cells that can generate either oocytes or sperm from a variety of other cell types, but that's not something that's not a stage that we're really at. And certainly not in a clinical option at the moment. So by and large, if you have a testis, then your germ cell is going to develop. If it does, it will be a sperm, and if you have an ovary, it will be an egg. I don't know if Evelyn and David agree with that.
Yeah.
Yes.
Okay. So a simple answer there then, really. So we'll move on now to the next question, which is from Pav Kaur. Apologies if I pronounced your name wrong. Is there a way to ascertain the number of eggs remaining in a female? Evelyn, I'll go to you first.
Well, there's an indirect measurement that's used clinically, and that's measuring a particular hormone, anti-Müllerian hormone, AMH, and that essentially measures or is an indication of the number of growing follicles, which is then an indication of the number of, or the population of non-growing follicles. So it's clearly not an accurate measurement, but it's an indication of the activity of the ovary.
Thank you. So we've got a number of questions. I was just conscious of the time, so I was just trying to think if I could merge any together, but I'm not succeeding. So we have one from Jan Rees. What might be the future options for treatment of POI, primary ovarian insufficiency, where diagnosis is made after the possibility of ovarian tissue preservation? I don't know, David, do you want to go on that one first?
Sure. Yeah, POII, premature ovarian insufficiency is one way to describe it. And there's another sort of version, premature ovarian failure, but these are there's possibly a genetic component to this, but I think the importance of the question lies in the fact that women who have achieved puberty may suddenly have a loss in their follicles within the ovaries, so they will go into a premature menopausal state in many ways, and Fragile X syndrome is one of the things that's been associated with this disorder. But the important thing from a fertility preservation point of view is that if you had this condition 20 years ago, you really had no options. Okay. And this happens within some populations to young women 18 years old, 20 years old, 21 years old, and other patients it can happen suddenly in their 30s. But for women who are before their reproducing years, so to speak, fertility preservation does offer an opportunity to preserve what's there. And of course, it also, when you look at kind of the economics of this situation, you're able, especially in women who are cycling, they can go through an ovarian stimulation cycle, and you can retrieve many eggs and freeze them away. Okay, so that even though their ovarian function may be destined to deteriorate over time, there's something to work with at a later stage of life. And this condition does not affect the ability of the uterus to accept and retain an embryo and sustain a full gestation either.
Is there anything you want to add on that, Evelyn?
Yeah, I mean, just to say clearly, as we understand it more, then there's a patient group that can be identified for the option to be offered tissue freezing. And that's not to say, I mean, transplantation could be an option. I mean, for example, the Turner's patients that do lose their eggs very early, they're now being offered tissue cryopreservation and subsequent transplantation. That has taken place in Edinburgh. Yeah, I mean, so it opens up more options, and the in vitro growth system would in the future also open up more options to these patient groups as well.
Something that's very dear to my heart, because I used to run the DAISY Network Premature Menopause Society. So yeah, it's be great. Now, I think we might just have time for one more question. There's a question which is quite a long one actually, but I'm going to go for it. Emily Walsh, and it's for Rod and Evelyn, and it's a two-pronged question, she says. So is there a reason why the focus of methodologies for fertility preservation are different depending on sex? Tissue transplantation seems to be preferable for males with less focus on maturation of stem cells in vitro prior to transplantation. Sorry, something's just popped up on my screen. I can't read the question. However, while ovarian tissue transplant has already resulted in live births, IVG, in vitro gametogenesis of oocytes, seems to be of more interest. Have there been difficulties in ovarian tissue transplant resulting in a shift of interest to IVG? Similarly, are there hurdles with culturing, differentiating sperm stem cells in vitro, meaning tissue transplantation is a more viable option? So if you can just manage to answer that in about a minute each,
Sure.
that would be fantastic.
Well, I'll start. I mean, clearly ovarian tissue transplantation has been going on for much longer than the testes. So, and it is a successful technique. And it's not the case, it's absolutely not the case that there's an interest in vitro growth because transplantation is not good. There's an interest in vitro growth from a basic scientific point of view, but also because not all patients can have the transplantation. So they store tissue and then it cannot be transplanted. But tissue transplantation is a successful technique and will continue to be a successful technique.
Brilliant. Thanks, Evelyn. Rod.
But I would just say that I think actually we're very aligned with the female. We're just 20 years behind. So I think the evolution will be exactly the same, that we will go for transplantation as the option that's most likely to work first. And that will then evolve into having a successful transplantation program, but also moving in towards other methods that might be useful in a wider population of patients, which would be those that then would need to benefit from in vitro maturation. So I think they're very much aligned. We're just a bit behind.